Growing better fresh produce. So we've heard a little bit about a farmer's perspective. I noticed that you're growing potatoes, which I thought was pretty impressive in a dry area. And then we've been talking about wheat. Well, I'm going to move away from wheat and I'm going to concentrate on vegetables, and I'm actually going to really narrow in onto leafy vegetables. We do quite a lot of work here at Harper in this area. So my title was The Link from Genotype to Crop Phenotype. And really I'm going to start talking about this impact of environment. And what do we mean by environment? I want to get my thank yous in first. Dave Pink can't be with us today. Paul Hand is, I think he's already in Spain for a Eucarpia meeting. Andrew Beecham is also there, but Yara is here today, and Justin Roberts is also in Spain. So it's unfortunate timing for us. And there's some other members of the fresh produce team last summer. I'm going to talk about breeding targets very briefly, just to set a scene. And then I'm going to just take a slide to define agronomy and remind us what agronomy actually covers. And then we'll talk about crop phenotype and how important that is to a farmer. Then I'm going to use a number of examples where I talk about how environment, and what do we mean by environment impacts on the phenotype. So there is a lot of pressure on leafy veg growers. There's pressure, we all know about environmental concerns, and every now and then news pops up about lettuces using huge amounts of pesticides. We've got big problems with price reductions. I'm not going to bore you with market data, but everything is down in price for the last year on year. Healthy eating campaigns, actually salads feature highly in these campaigns, and some particular lines where maybe more on the Brassica side of things are put forward as a good option. There's also food safety worries, and we're not really going to cover that today, but that is something that can also be dealt with from a breeding perspective. So is genetics the only solution to these challenges? And it's very easy to sit back and say, oh, well, we'll find a genetic solution. We just need to get a bit of breeding there, everything will be sorted. But I want to put forward the argument that agronomy has a part to play as well. And so let's, when I talk about agronomy and I think of Harper students want to be agronomists, what that means is they're going to make recommendations for pesticides and to a certain extent fertilizers. Agronomy is more than that. It's a definition from the Oxford English Dictionary, the science of soil management and crop production. And then I've done the ultimate no-no. I've gone to Wikipedia, which we advise our students not to do. I've got quite a nice broad definition there. Agronomy is the science and technology of producing and using plants for food, and encompasses work in the areas of plant genetics, plant physiology, meteorology, so really the weather, and soil science. So actually, agronomy covers a lot of the things that we're doing to crops when we get to the field. So we're interested in maximising the phenotype, and we just heard a little bit about this debate about genotype versus phenotype. So you're all familiar with phenotype. It's the set of observable characteristics of an individual plant resulting from the interaction of its genotype with the environment. Now we group that interaction together in the equation. It's additive, genotype and environment, and it's also an interaction of genotype times environment. And that one is a particular concern when we start thinking about phenotype in changing environments. And that can be changing in terms of spatial, so it could be spatial heterogeneity in a field, or it could be temporal heterogeneity, so over a growing season. And remember, agronomy is what a grower does with the genetics in the seed they buy. The genetics are fixed, it's in the seed. Agronomy is their ability to manipulate the environment that the plant is growing in, and consequently manipulate the phenotype. So what is the phenotype? Well, it doesn't take long to go on Google Scholar or Web of Science and put in plant phenotyping and get a nice image of Arabidopsis being phenotyped, or any other model plant species. And it's great stuff. We're looking the differences between all these lines are accounted for by the genotype, because these, in this example here, are being grown in standard conditions. The environment is equal for all the plants. And we do this sort of stuff here at Harper, and here's some lettuce transplants that we've put out, and we're looking to see how they behave in an environment that we've got some sort of control over. The challenge we have is that growers are not functioning at that level. They're functioning at a field scale. So for a grower, the growers see the crop phenotype, not the plant phenotype. And on the left here we've got some spinach being grown in Nottinghamshire, and you can see two different plantings here, very pale here compared to here. I don't know whether that's a variety difference. I don't know whether this has got base fertiliser in and this hasn't. I don't know, but it looks different. Those plants are going to be doing different things. And here in the fens of Cambridgeshire we've got some whole head lettuce. Again, the grower, when he's looking to see has he made money, it's off the field, not per plant. Now Dave Pink, who can't be here with us today unfortunately, put a paper in New Phytica, and he talked about this sort of crop phenotype in terms of actually disease resistance. And he said deployment of desirable genes will have to be integrated into a whole crop management approach, focused on achieving the desired crop phenotype. And it would involve pathologists, agronomists, breeders and farmers. And he termed this phenomics. Now phenomics has gone on to mean something slightly different. It's much more high throughput phenotyping systems now. But this is what he defined as phenomics at the time. They'd be informed by the results of genomics, etc., but would take into account the most important factor for farmers and growers, and that's economics, because they want to make money from the plants that they grow, and they make money on a field scale. So we need to optimise the growing environment, agronomy, and combine this with optimised genetics for stress resilience to get the ideal crop phenotype. So I'm going to just focus initially on agronomy. Now, fields are not uniform environments. And this is something I stumbled across a few years ago on Google Earth. And this is up near the Cambridgeshire Norfolk border, a place called Southery. If you've ever been over that way, this is absolutely superb growing land. These are peats, black peats, so up to 40% organic matter, which is off the scale when it comes to how good is your soil. The difference is, of course, when you look at it, it's drained land. And you can see in this map, you can see where the old waterways went. Now in some fields you get a whacking great slab of this, and they call them rodents when you go over that way, but on some fields it's actually quite small. But these fields, when you stand there, you think, wow, this is a really good field, and they're superb, but they're not uniform. So actually this means this E in this interaction is changing across a planting. How can we manage that to make it uniform, to get a uniform crop phenotype? Well, if you go to a grower, they'll say, well, over in that corner it's usually a bit wet and a bit smaller. Over there they can be a bit bigger. They can work at that level. It's not very scientific. You could start taking soil samples, digging soil pits, quite time consuming, back-breaking work, and will only give you information quite specific to the area that you've actually dug the hole. You can go to yield maps. People will tell you the yield they take off a field in terms of wheat, but more and more now we're seeing combines running with sensors on them that are actually mapping the offtake from the header, from the yield going across the field, and generate maps, like heat maps. So there's a lot of crop in this area and there's less crop in this area. We're moving forwards now to people actually going with soil maps, and they use apparent electrical conductivity scans, or NIR, and these will actually give a sort of a proxy measure for differences in soil texture over the field. And Yara Boo-Boo, who's a PhD student here at Harper, is doing this, and she has a poster if you want to have a look, I encourage it, in the main hall. But she's been able to take data, and here we've got a field. This is the EC map, a heat map if you want. This is low EC is the yellow, moderate EC is this strange green colour, and high EC is the dark green colour. And if you go in and you start digging soil cores out of these areas, you're getting different soil textures. So immediately, Yara is able to go in, scan the field, this is commercial services being provided, and identify different sorts of soils in that field. And when you go and look at the plants in those areas, you'll see the plants are growing differently. They're still growing well, but there's some variation. So in the low EC area, lots of weeds. We can see quite a bit of soil because we have smaller plants. And if you go into the moderate EC zone, few weeds because they're being competed out, maybe. Larger plants, good crop cover. And when we get through to harvest, we find that, and for those who are not familiar with a lettuce plant, effectively you leave half the plant behind because you go in and you cut the head out of the lettuce plant. So if you look at the total growth of the plant, the total biomass, you're finding that in this low EC zone, around about 10% reduction in total biomass per plant. When you then look at that from a marketable yield, that difference, that disparity is still being carried through even following trimming. Now, this is still a very commercial sellable head, but that's a significant statistical and significant commercial difference for that grower. So you've got quite a bit of variation across the field. From a grower's perspective, they want to minimise that. So how can we do it? We can understand the causes of variability, understand the variation in E accounted for by the field environment. We don't know how to do that at the moment. We don't know what is really going on, and you're always having a go at that. We need to then find out the critical ranges. So if this is high and this is low, do we need to start treating these zones differently? We don't know the answer to that. We now have the techniques to start managing fields in zones, and the precision equipment is there. We can start applying water in specific areas. You can go in with variable rates of fertiliser. You could even change your planting rates, so where you've got lots of resources, you could actually put plants in at a higher density. But the target would be to reduce variation within the crop, and that would mean reduce E within that equation for phenotype. But the other things, and Guy spoke very well about climate change, but climate change can be long-term and it can be short-term, or climate differences can be short-term within a growing season. We can have a wet season, or we can have a season that starts wet, finishes dry, or starts dry and finishes wet. So we can have dry, we can have cold, we have cold snaps early on in crops or late in the crop season. We can have weeds, and nettles is always a really big problem for lettuce growers. Diamondback moth, especially for Simon leather. So we have pests coming in and we have diseases coming in. So the growing environment, this E component, changes over the season as well. So what can growers do to mitigate the stress? Because what we're really talking about is environmental stresses, moving away from the ideal condition. Well, biotic stresses, weeds, pests, diseases, actually there's a reducing, and we'll talk about it at the very end, but there is an armoury available to growers. For weeds, they could mechanically weed, or they can use herbicides. For pests, they can use specific insecticides. For diseases, you've got options there with certain pesticides, but also some cultural controls. Grow your lettuce in a tunnel, for example. What I'd say, though, for abiotic stresses, it's much harder. So actually we can get periods of really hot weather in the summer where plants will go into thermodormancy and just stand still. We can't cool fields, so we can't respond to that. The grower can do nothing in that environment. They just have to sit back. Cold, at certain times early in a crop's life, we can put crop covers over. So put a thin blanket if you want, put a fleece over the top. You might do that at the start and the end of the seasons when it's getting a bit colder in your plantings. But as the plants get bigger, you can't do that. Well, it's quite easy to get your head around what happens if it's droughted. You put the irrigation on. But what happens if you get a wet period? Well, maybe you've prepared your soil in such a way it drains a bit more freely. You might be able to mitigate with nutrients being flushed away by adding additional nutrients, but you're quite limited there. And then we sometimes forget that we do get cloudy periods and sunny periods, and if they come at critical times in crop development, you can't do anything about it. You just have to sit back and let that happen. And the challenge is that these aspects here, these environments, these E's in that phenotype equation, there's actually very little that can be done by a grower. So then we need, in that case, to be looking to the genetics, and we need optimized genetics for stress resilience to allow us to get the ideal crop phenotype. Now, with apologies to Jim, I'm going to completely ignore pest and disease resistance or tolerance. I'm going to completely ignore biotic stresses. And I just want to focus on a few examples of abiotic stress situations. So let's think right back. When we put lettuce in the field, we've got transplants going in, and transplants are actually really sensitive to stress because you've got a plant with maybe 3 or 4 true leaves on it. You've got a little block which contains some water, a little bit of nutrients, and you've got a root mass there that's waiting to get out into the soil. But this is the sort of environment they go into. It's a bit cloddy, it's on an angle, it's not got great contact, and actually in quite a short time, the moisture in the block can be gone, and actually you could start to struggle. So the ideal sorts of transplants have been those that root deeply, quickly. And it's been worked by Kertiriou in Holland has shown that deeper rooting transplants, this was in lettuces, are linked to stable field performance across trials, locations and season, and may constitute a trait for robustness for lettuce. and they were showing that some varieties, for example Matilda here, which is a Dutch variety, did a lot better job than Mariska in rooting through the profile. So I bet you haven't seen a pint glass of roots before. We have a PhD student, Justin Roberts, here joint with Nottingham, where he's actually looking to set up some fairly high throughput phenotyping assays for lettuce rooting from modules. And this is a plastic pint glass he puts inside a black sleeve so he can quickly get the roots in 3D dimensions, picks it out, and is working on some imaging technologies that can be used. So rooting traits are going to be really important. But can we identify genetics for more sort of general abiotic stress resilience? Because stresses rarely come on their own. So high light levels are going to be when it's warm, but it's probably going to be a bit cooler when you've got lots of rain. You might have drought when you've got higher temperatures. So what can go on there? Well, there's some work we've done joint with Warwick in the vegging program, where we're looking at actually young Brassica plants in the transplant stage, and we were stressing them, so giving them periods of cold, sort of a cold shock. They're being mollycoddled in a lovely, nice, warm environment, give them a cold shock for a few days, bring them out again, or alternatively drought them so they wilt and then re-wet them up and grow them on. And you can see there's different response patterns, but the ones that are most interesting are those which don't have the asterisks, because it's not rocket science to say that if you drought plants they are smaller. But actually here we've got two lines, and particularly this one's of interest, where it's been droughted to wilting, but when it's grown on again it actually hasn't led to any yield reduction in the young plant. So what is it doing? We've got to look into that in more detail. And finally, with environment, there are some problems with lettuces particularly, which are caused by environment but are really hard to breed for because we're unable to reproduce the conditions routinely. So this is tip burn, big problem for growers, and so ideally we want to be developing resilience to tip burn. Now, tip burn is erratic, so the symptoms occur erratically. We know that it has a big effect on plantings. It can write off large amounts of crops. And we have some understanding of the causes. It seems to be to do with the flux of calcium to the leaf margins. So any conditions that lead to reduced transpiration, such as waterlogging or drought, which seems slightly counterintuitive, but anything bad really, poor rooting and high relative humidities will lead to tip scorch. Now, if you want to try and breed for that, that means you have to be able to repeatably generate those symptoms. And therefore, the resistant phenotype can only be expressed in a conducive environment. So in a way we can actually then take that E from the G times E interaction and play with the E, and we can develop hydroponic systems, and that's what we've done here. For 20, 30 years people have been trying to get repeatable tip burn symptoms in lettuce, and it's not been possible. So last year Andrew Beecham, who's a postdoc at Harper, managed to optimize a system where he can get about 90% symptom development in single lines. And so we've gone to the GRU unit at Warwick, and also some stuff from Holland to get diversity set together, and that's the setup when it was planted by Andrew for one of his plantings, and that's what it looked like last week. And you can see a wide range of morphologies. Here we have the wild types, we've got some more cultivated types, we've got open hearts, we've got closed hearts, we've got loose leaf types. And so Andrew's able to go in there and start scoring for expression of the phenotype, and the phenotype is undesirable by a grower. And we can get a range of symptoms, and this again, we took these pictures last week, and we see on different lines we're getting different levels of tip scorch. This is a nightmare for a grower, and this could just about be bearable. So it allows us to start breeding for these erratic problems. So in my short time I've tried to say we need to optimize the growing environment through agronomy, and we need to combine this with optimized genetics for stress resilience to get the ideal crop phenotype, which allows us to have consistent yielding over fields and over time. But there's an elephant in the room. The scope for agronomic inputs is reducing. And Guy was telling us about the loss of active ingredients. So that's going to have a major impact on biotic stresses. You'd expect to see greater biotic stresses. And if you start talking about water use, again, Guy really nicely pointed out the fact that there's going to be challenges irrigating crops in periods of drought. If the climate models are correct, we would expect to see more extreme weather. So we could have flooding as well as droughting. And there is also on top of it the issue with cost, because if inputs become more expensive, then growers become more reluctant to use them unless the price of the product goes up. So actually, I'm quite happy to be saying this in front of plant scientists, actually we need more stress resilient genetic work, because this is probably going to be the way forward. Because even though I stand here with a pair of, I suppose, sort of, and metaphorically speaking, a pair of muddy boots on, actually the reality is that what an agronomist can do is going to be reduced in some areas, and we're going to need to rely on the stress genetics. So in summary, agronomy, is it the link between genotype and desired crop phenotype? Yes, where you've got the ability to manipulate it, and no, where you haven't. So we can reduce environmental variability within a field, and between plantings to a certain extent. We can use agronomy to interact with the best genetics to allow us to deliver the optimum phenotype, but that phenotype may need to be expressed in varying environments. We're going to have harsher growing environments, which means that's going to become more difficult, and we need to identify, I think, not only biotic but abiotic resilience that can be utilised in the breeding germplasm. Thank you very much.